Once gametes have been produced, they can go on to participate in a fertilization event. And of course, this is important in all of our diploid life cycles of our animals and our plants. In general, our plants and animals are going to be our diploid reproducing organisms. Some of our fungi have a diploid part of their life cycle, and so it's going to be similar to plants and animals as well. Because we're in genetics and we usually focus on plants and animals, we're going to go ahead and just assume we're talking about humans here. And we're just going to go through the cycle of fertilization just for a very quick review. So meiosis happens in either the ovaries or the testes. And we've got our haploid gametes that are produced there. So meiosis happens if the cell is produced in the ovary, it's going to produce an egg. If it's produced in the testes, it's going to produce the sperm. So notice here that our egg is rather large. It's a very large cell, very large nucleus, and then the sperm is very small. Very little cytoplasm comes with the sperm. The sperm is basically just the chromosomes at this point, whereas the egg has cytoplasm and the mitochondria that is going to provide all of the energy for this newly growing organism. Each of these are haploid, so of course when they come together during fertilization, we restore the diploid state. Then mitosis takes over. Mitosis, remember, is for growth and repair and replacement. So we're going to assume growth here. That cell's going to begin to divide by mitosis into the full-grown organism. And then, of course, meiosis happens in our gametes. The process of the production of gametes is called gametogenesis, and then our process of producing sperm specifically is called spermatogenesis. Egg production is called oogenesis. We pronounce this genesis. When we learn about meiosis, we are learning about a generalized process where we start with one cell, divide into two, and then each of those divide into two more for this overall production of 4 cells. And the gametogenesis of sperm is essentially that. We're going to get basically 4 identically shaped cells at the end, each with their own haploid complement of chromosomes. Oogenesis is a little bit different. At the end of oogenesis, we actually only get one, I would say, functional egg that can be fertilized. And so we're going to talk about the differences. Now, the differences here are actually really only in their vocabulary. And a lot of learning what's going on in gametogenesis is really learning the vocabulary words. So before you do anything else, I would make some flashcards here and just really get the terms down, because it's going to be the terms, I think, that trip you up instead of the process. For the process of spermatogenesis, we are going to start in the testes with a cell called a spermatogonium. Now, this gonium suffix is kind of a term that means the original. So the spermatogonium is going to be the original sperm cell. Now, this is a set of cells, and we see a similar set in egg production as well in the ovaries. This is a set of cells that goal in life is to be ready for meiosis. And these cells are maintained by mitosis because we always have to start with 2N. So our spermatogonia continually reproduce by mitosis. And when they are ready to undergo meiosis, they flip into a cell type called a spermatocyte. Now that spermatocyte is what is going to undergo meiosis. So spermatogonia is kind of the stockpile of cells. And when one of those cells is ready to undergo meiosis, it flips from a spermatogonium, or yes, spermatogonium, into the spermatocyte that can now undergo meiosis. So that first cell that kind of flips the switch that's ready to go into meiosis is called the primary spermatocyte. So our spermatocyte is the one that is actually going to divide and eventually become the sperm. That spermatocyte is diploid. And remember, our end result is haploid. So that spermatocyte is going to go through 2 rounds of meiosis. We're going to end up haploid. So meiosis 1, that primary spermatocyte undergoes meiosis 1. You know what happens there. We reduce our chromosome number, and now we have 2 spermatocytes. Those 2 spermatocytes are called secondary spermatocytes. So we've got the first round of spermatocyte and the second round of spermatocyte. So those 2 spermatocytes are now haploid. This is just the end result of meiosis 1. Then those spermatocytes go through meiosis 2 and become spermatids. Now I think of spermatids as kind of baby sperm. They are the end result of meiosis 1, but they're not mature sperm yet. They are going to go through a maturation process, and each one of those spermatids is going to become sperm that can then fertilize the egg. So my advice here is flashcards and draw out what's happening. Flashcards, flashcards, flashcards. Once you get this one down, then oogenesis is really easy. And again, you know the process here. This is just the process of meiosis that we've already covered. We're just adding in names to the cell types for each step. Oogenesis is going to follow the same general process of spermatogenesis. So I would suggest, if you have not got a grasp on spermatogenesis yet, that you go back and review that and at least get a good understanding of the outline. Remember that this is just meiosis. We're just giving our cells some new names here. All right, so we are going to start with oogonium, which are our stockpile of cells in the ovaries that are maintained so that individual cells can undergo the process of meiosis when they're ready. In females, all of the eggs are produced before the individual is even born. They are produced in utero, and then they get sort of stalled during meiosis. And then when the female reaches sexual maturity, she's going to start releasing eggs. And meiosis is actually going to finish upon fertilization, which is a little bit strange. We aren't going to go into that too much here. But just keep in mind that eggs are actually produced way, way back in utero during development. Sperm are produced during a lifetime. This is kind of why females hit menopause, and there's a point where they basically sort of run out of eggs, or they at least stop releasing them, whereas males produce sperm forever. Okay, so we've got our oogonium. One of the cells in the oogonium is going to flip into meiosis, and it becomes that diploid primary oocyte. I'm going to walk through the whole process, and then I'm going to come back and I'm going to talk about a couple key points that are a little bit odd compared to spermatogenesis. So that primary oocyte is going to split into 2 cells. One of them is called the secondary oocyte. The other one is called the first polar body. And if you look closely, you'll notice that that secondary oocyte is a little bit bigger. That first polar body is a little bit smaller. What happens here is there is an unequal division of the cytoplasm, because the end result is going to be that really large egg that is filled with basically energy-rich, nutritious cytoplasm. And so we need to get that cytoplasm from somewhere. And so one of these cells, one of these, what would be the oocytes, is basically the side effect of that. It is a little tiny cell that is basically just chromosomes. There's hardly any cytoplasm in there, and that cell is called the first polar body. That polar body is eventually going to disintegrate and basically just be broken down and not used in the body. That secondary oocyte is the one that is going to undergo the second round of cell division and what is eventually going to become the egg that is fertilized and released. So when that happens, we get another cell division, we get another unequal division of the cytoplasm, we get a second polar body that is going to disintegrate. Again, that is so that the other cell, the one that is going to be the fertilized egg, gets all that extra cytoplasm. Here we have something that is very different from the sperm. The sperm just undergo their primary oocyte division into secondary, and then secondary oocyte into spermatids, and then they're done. For females, meiosis is arrested during development, and meiosis doesn't continue until that egg is released and fertilized. Now, in some animals, and you can see the two little bubbles here, in some animals, the meiosis happens when the animal is in heat or she's reached sexual maturity. The egg finishes meiosis and then becomes available for fertilization. In humans, the second division doesn't actually happen until the egg is fertilized by the sperm, which is kind of weird. So the egg is fertilized by the sperm, it triggers the second round of meiosis, and then the polar body is released, and then the egg sort of finishes meiosis at the same time that it is fertilized. So the chromosomes finish separating, that polar body is released, and then that egg is fertilized, and we get the fertilized zygote. This slide here is just the remainder of the description of the steps so that you can get those in your notes.